Okay, hi. My name is Asja. I'm from the physics department, and today I'm going to be telling you about patterns on pollen and how they're a beautiful consequence of biophysical forces at microscopic scales. Okay, so there are about 250,000 species of plants that produce pollen, and each individual species produces pollen that has its own unique pattern on the surface. So there's a huge diversity of different types of patterns that are found in nature on these pollen grain surfaces. And crazily enough, no one knows either how they form or what they're for. So in my thesis work, I wanted to try to tackle these two questions. So in order to do that, we first have to understand what is known about pollen development in the literature already. And it's known that all species follow the same set of fundamental developmental steps. So all pollen starts off in a plant organ called the anther as a single cell that goes through meiosis. And then a really interesting step happens where each individual cell produces a material that's a mixture of different types of sugars, and it deposits it on top of its cell membrane. And then underneath the cell membrane, underneath this sugar mixture, because of the pressure of the sugar mixture, the cell membrane starts to wiggle and undulate, sort of in a way you can think of like a grape turning into a raisin. After this, there's a new material that's deposited. It's called the exine. And it's the exine that we actually see when we look at a mature pollen grain under the surface. But because we were interested in understanding the pattern formation step, we wanted to actually understand this step first. What exactly was happening with this sugar mixture and the underlying cell membrane? So in order to do that, we spent months doing our own microscopy. And what we found was that actually this sugar mixture on the surface was phase separating. And that was actually causing the pattern to form. So the way it does this is you start off as a uniform mixture of the surface, of the material on the surface. And then, just like oil and water, if you leave them to their own devices, they'll phase separate. In a similar way, the different types of sugars in this mixture were starting to phase separate. So that's what you see in the middle. And on the end, the really dense sugars were then patching the surface, and they were pushing down on the cell membrane, and that caused the pattern to form. We know this is the case because if you now compare this final step of development to a mature pollen grain, you can see that wherever you have this dense sugar, that's where there's a hole. And then the exine, the material that's deposited last, gets deposited around these holes. So at this point, we figured out that it's actually the prime exine sugar mixture phase separation that templates patterns on pollen. So what we next want to do as physicists was try to create a model for this. So what we did was we developed a model for a phase transition from a uniform sphere to a pattern sphere. And as I said, left to its own devices, this pattern sphere will come out because it's the lowest energy state. With this model, we had a couple of parameters that we could sort of tune and tweak to find out all of the different patterns that our model can predict. And then we wanted to compare them to actual pollen images to see if this model is applicable or not. And we did that, and we were really, really happy with the results because, as you can see, each of these columns on the left has real images of pollen grains, and on the right, these are my simulations that match those patterns. And you can get things like spikes, you can get things like stripes down here, and then my very favorite is that one up there. I'm not supposed to leave this box. Okay, so at this point, we actually noticed that only about 10% of pollen is actually this really nice ordered, symmetric state. The rest of the 90% look a little bit more disordered, but we also wanted to be able to understand those. So we went back to our theory, and we now looked at the phase separation from uniform state to a pattern state, but we stopped it somewhere in the middle. And this can happen biologically if you have a second chemical that comes in and binds the sugars together so they can't fully phase separate. And when we did that, we were able to find these types of patterns which really do recapitulate the rest of the 90% of the patterns that you see on pollen grain surfaces in nature. So at this point, we have figured out that the pattern formation mechanism is a phase separation of a sugar mixture on the surface. We also have a way to mathematically describe practically any surface that we can find on a pollen grain. And the last thing we want to do is see if there is a purpose to these surfaces. So in evolution, usually the way it works is that if a particular feature has a function, evolution will select for it. So we were assuming that we'd find that any sort of smooth state like this at the bottom would eventually evolve into a pattern state because it wanted to have that pattern because that pattern was favorable. We were really surprised to find the complete opposite. Actually, they evolved from patterned states to smooth states. So evolution was actually selecting for these smooth, unpatterned pollen grains. So our conclusion to this was, actually, maybe these patterns don't have a function, and maybe they're just a natural consequence of the phase separation process. So this actually told us a little bit about ourselves. As humans, we tend to look for patterns in the world around us and ascribe function or meaning to them. And nature doesn't actually have to work that way. So this is one of those cases in which, even though we gravitate towards, these really beautiful symmetric states, they might just be an accident of nature. So the next time you have allergies, I can't help you. This research doesn't do anything for that. But hopefully you'll find, hopefully you'll be at least excited by the fact that they're beautiful under a microscope. Sorry.